The overall goal of this methodology is to identify multiple phosphorylation of an intrinsically disordered protein by nuclear magnetic resonance spectroscopy using tau protein as a case study.
This method can help to answer key questions in molecular regulation related to disease, such as the impact of the phosphorylation on the protein interaction with its molecular partner.
The main advantage of this technique is that we can identify site-specific phosphorylation in a disordered protein and link it with structural and functional changes.
So this method can provide insight into the function. It can also be applied to other proteins, such as the disorder domains of the NS5A protein from the hepatitis C virus.
The implications of this technique extend toward therapy by development of protein-protein interaction inhibitors.
This method will be demonstrated by Clément Danis, Clément Despres, Luiza Bessa, and Hamida Merzougui, the PhD students from our group.
To begin, gently mix 50 microliters of competent BL21DE3 cells with 100 nanograms of plasma DNA, forming 10 to 50 million colonies per microgram of plasma DNA in a 1.5 milliliter plastic tube. After placing the cell mixture on ice for 30 minutes, heat shock it for 10 seconds at 42 degrees Celsius. Then place the tube back on ice for 5 minutes before adding 1 milliliter of room temperature Luria-Bertani or LB medium. Incubate the bacterial suspension at 37 degrees Celsius for 30 minutes under gentle agitation.